All right, so welcome back. So now we are starting our discussion of the membrane potentials. And the membrane potentials are going to be the electrical changes that are taking place in the plasma membrane of the nerve cells that allows it to conduct and create an electrical impulse. So if we're looking here at a regular cell, like any cell in the body, inside the cells, we've got much more negative charges than outside of the cell membrane. So inside your cells, in the intracellular fluid, you've got more negatively charged ions compared to the outside. Outside, I will have more positively charged ions. So this will create a difference in the membrane potential. What this means? This means that electrically, inside is more negatively charged if you compare it to the outside. So if I'm measuring the difference between the inside and the outside of the cell membrane by a voltmeter, I will find that inside is more negatively charged by value of 80 millivolts. So when I measure the difference between inside and outside, I will find that inside is more negatively charged than the outside by 80 millivolts. So the difference in between outside and inside is going to be negative 80 millivolts. Another thing is we need to consider here when we discuss the membrane potentials that outside I have much higher concentrations of the sodium. This is the most prominent cation on the outside. The most prominent positively charged ion on the inside of the cells, it's the potassium. So inside and outside, you have both negatively and positively charged ions. But inside, I have more negatively charged ions. That's why inside of the cell membrane, it's going to be more negatively charged than the outside, which is more positively charged. So what do I mean by conducting an electrical signal? It simply means that I will be changing the membrane polarity. Rather than outside being more positively charged and inside is more negatively charged, I will flip the charge. Inside will become more positive than the outside. By this means that this cell is conducting an electrical signal. How this takes place? In response to a stimulus. And the minimal stimulus, if you remember, it's called the threshold stimulus. If I've got a threshold stimulus that triggered this cell to send an electrical impulse, this cell is going to open enough sodium channels Those are my sodium channels on here. They have two gates, an outside gate and an inside gate. Inside gate during rest, it's gonna be open, and the outside gate, it's gonna be closed. Whenever I receive enough stimulation to this nerve cell, the outside gate, it's gonna open. And now the sodium having a high concentration outside of the cell is going to be traveling down its concentration gradient. So inside the cell, I have much less sodium compared to outside of the cell. So the sodium, as the channel is open, sodium will keep moving to the inside of the cell. So what allowed the sodium to move inside the cell? First, the high concentration of sodium outside of the cell compared to the inside of the cell. Another thing is that inside of the cell is more negatively charged. So, and sodium is more positively charged. So what's going to happen? The sodium is going to be attracted to the side with negative charge, means it moves from outside of the cell to the inside of the cell. As more sodium keeps moving in, if I open enough sodium channels, this will cause a cascade of sodium channels opening to the point of no return. So the sodium keeps moving inside the cell, and the inside of the cell, as a result, is going to become more positively charged. So the inside is going to be becoming more positively charged. So here I am losing my membrane polarity. So inside here turns to become positively charged compared to the outside, which will become more negatively charged. And we call this state, it's the depolarization phase. Until when? Until I reach 30 millivolts. So inside becomes more positively charged by 30 millivolts. At this point, the sodium channels gonna close. So the internal gates now are gonna be closing. And no further influx of sodium, it's gonna be taking place. Outside, I still have high sodium concentration compared to the inside. But the sodium is not gonna be traveling inside the cell when I reach 30 millivolts, simply because those channels gonna be closed. Inside here, it's still more positively charged compared to the outside, which will became more negatively charged. So in order for me to send another signal along this nerve fiber, I need to return back to my resting membrane potential. I need to return back to my negative 70 millivolts or negative 80 millivolts. So what's going to happen? you can't simply get the sodium that did move inside out. Why? Because you're going to be trying to move the sodium against the concentration gradient. So it requires energy, and it's going to be consuming time. You need to do this in milliseconds. So I can't allow the sodium out at this point because simply I have higher concentrations of sodium outside of the cell compared to the inside of the cell. So I would be trying to move the sodium against the concentration gradient. So how can I return the inside to become negatively charged another time for me to be able to resume sending my electrical signals? In order for me to do this, rather than returning the sodium out, I will instead find another positively charged ion that I have high concentration of it inside my cell. And this is going to be my potassium. I will open the potassium channels when I reach 30 millivolts. And the potassium is going to be moving from its high concentration gradient within the cells to its low concentration gradient outside of the cells. So potassium is going to be leaving the cell. And as I lose positively charged ions to the outside of the cell, now the inside of the cell will return back to its negative state. Compared to the outside, will be returning back to its more positive state. So let me modify this here. So according to the textbook here, it's a 70 instead of 80. So let's stick with it as 70 millivolts. As we're talking about the nerve fiber, not the muscles. All right, so inside returns back to its negative state. So the inside will become more negatively charged by a 70 millivolts. So the inside now is going to be negative 70 millivolts. So you see on here, I reach 30 millivolts. Looking here at the PowerPoint, 30 millivolts. What happened at this point? I closed sodium channels. And I opened potassium channels. And when I open potassium channels, the potassium here gonna be leaving the cell. Potassium is gonna leave the cell, and the cell will return back to its negative state. So now I've reached negative 70 millivolts. This phase is what we call the repolarization. So I started by the resting membrane polarity. Inside is negatively charged compared to the outside. It's more negative than the outside by a value of 70 millivolts. This is during rest. This is my polarized state. I received a threshold stimulus. And when I receive a threshold stimulus, the sodium channel is going to be opening in a cascade pattern. Until when? Until the inside of the cell becomes more positively charged than the outside. Not only this, but it will reach 30 millivolts. So at this point, inside is more positively charged, outside is more negatively charged. This is a state of depolarization. I have lost my polarity. I want to return back to my polarized state for me to send out another electrical signal, to keep sending the electrical signals. What is the fastest way to do this? Rather than trying to get the sodium out against the concentration gradient, what you're gonna instead do, you're gonna be just closing the sodium channels to prevent any more influx of the sodium, and you're gonna open potassium channels. As the potassium moves out, the inside of the cell membrane will become more negatively charged, returning back to your resting membrane polarity. I will return back to my negative 70 millivolts. But as those potassium channels are going to be slow to close, I will keep leaking potassium out. Even after I reach my resting membrane potential, after I reach negative 70 millivolts, I will still be leaking potassium to the outside. So the inside will even become more negatively charged than it was at the resting state. So now the membrane potential is lower than the resting membrane potential. We call this as a hyperpolarization. I'm even more negatively charged than my resting membrane potential. Did any of this required energy? Did any of this required energy? Did you consume any ATP to allow the sodium in, to allow the potassium out? No, you didn't. You didn't consume any energy. So how come your brain needs a continuous supply of oxygen and glucose for it to function? So where is the energy going? If the main function of the brain is to conduct electrical signals, and the electrical signal creation and conduction doesn't require energy, so how come we're saying we need continuous supply of glucose and oxygen for the brain to function? So where is this energy is getting consumed? As you're saying here, Theodore. So if you kept doing this all the time, over and over and over, what's going to happen? If you kept moving the sodium inside the cell, and you kept moving the potassium outside of the cell, so over time, what's going to happen? You're going to be disturbing this difference in the concentration of those ions. So over time, if I kept moving sodium inside the cell, the sodium going to be building up inside the cell until I reach a balanced state. So inside the cell, I have the same concentration of sodium as outside of the cell. So no more sodium going to be traveling in. And if I kept putting potassium out over and over, what's going to happen? Outside potassium levels going to be increasing to the extent where I won't be able to allow any more potassium to move outside of the cell. So actually the energy required by the brain is going to be used to maintain high concentration of sodium outside of the cells and high concentration of potassium inside the cell by using something that we've discussed back in chapter 3, something called the sodium potassium ATPase pump. So running in the background, excuse me, running in the background all the time, what we're doing, we are activating, we are consuming ATP to move the sodium from inside of the cell. We move 3 sodium outside, and we bring 2 potassium in. So if you remember, sodium, potassium, ATPase pump, it's binding to 3 sodium, it's breaking down an ATP, and it will rotate. It's going to be moving the sodium outside of the cell against the concentration gradient. You see on here, you still have high concentration of sodium outside of your cells compared to inside of the cells. I am moving the sodium against the concentration gradient. And at the same time, what I'm doing here for the potassium, I still have high potassium levels within my cell compared to outside of the cells. I have much lower concentration of potassium. I am moving the potassium against the concentration gradient. So again, why the brain needs continuous supply of oxygen and glucose? Why do I need high energy? Because simply running in the background is going to be the sodium potassium ATPase pump that allows you to keep sodium at high level outside of the cell and potassium at high levels inside the cell to maintain the difference between the outside and the inside in terms of sodium and potassium concentrations. That's why all the time you're going to need ATP, all the time you're going to need energy, because if you only are doing this, moving sodium outside of the cells and potassium, moving sodium inside of the cells and potassium outside of the cells, what's going to happen over time, you're going to lose this difference in the concentration gradient. So you need to have the sodium potassium ATPase pump running all the time in the background for you to maintain this difference in the concentration gradient between outside and inside. All right, any questions? Any questions? Does this make sense? Does this make sense? All right, so let's watch this video together. All right, so you see on here, the action potentials are going to be transmitted between the nerve cells. The axon hillock is where I've got the summation of those signals for them to be conducted along my axon. The membrane of the axon contains those channels, as you see. I have the sodium and the potassium channels. Those are called voltage gated. They will be activated according to the voltage of the membrane. If we're measuring the difference between inside and outside, we will see that inside the cell, it's going to be more negatively charged. If I've got enough stimulation opening the sodium channels, the sodium going to be traveling to the inside of the cell, and it will keep opening in a cascade pattern once I reach enough stimulation. More voltage gated sodium channels going to be opening, allowing more sodium to travel inside the cell until I reach 30 millivolts. At this point, I will be using the inactivation gate of the sodium channel to close the sodium, and in the same time, I will open the potassium channels. And the potassium going to be leaving the cell, bringing the inside of the cell membrane to return back to its negative state. But as I keep the potassium channels leaking potassium to the outside, even beyond the resting membrane potential, I will reach even a more negatively state, which is what we call the hyperpolarization. All right. Questions? All right, so during the resting phase, only leakage channels of sodium and potassium are open, and all gated sodium and potassium channels are closed. In the depolarization phase, I will allow the sodium influx by opening the voltage-gated sodium channels. And as I allow the sodium influx, now the inside of the cell membrane will become more positively charged, allowing the depolarization phase of the action potential. In the repolarization phase, when I reach 30 millivolts, I will be closing the sodium channels. This will decline the membrane permeability for the sodium. And at the same time, I open the potassium channels. So if we look around here, closing sodium, opening potassium. And as I open the potassium, potassium is going to be moving down its concentration gradient, moving from inside of the cell to the outside of the cell. This results in restoring the membrane negative state. As some potassium channels going to remain open even after I reach the resting membrane potential. This creates a hyperpolarization state. The cell membrane is going to become more negatively charged than its resting state. See on here, started by opening the sodium channels. When I reach enough stimulation, influx of the sodium can result in the depolarization phase of the action potential. When I reach 30 millivolts, you close the sodium channels, preventing any further influx of the sodium, and you open the potassium channels, allowing the efflux of potassium outside of the cells. Losing potassium out and closing the sodium can result in the repolarization of the cell membrane. So my cell membrane here returns back to its resting membrane polarity. But as some of the potassium channels gonna remain open even after I reach my resting membrane polarity, the cell membrane will become even more negatively charged on the inside than its resting state, and we call this is my hyperpolarization phase. So for the third question for the in-class activity, for today. What is the resting state of a neuron? What do we call the resting state? Depolarization, resting membrane potential, repolarization, hyperpolarization, or action potential. What's the resting state of a neuron? It's my resting membrane potential, exactly. Some things that we need to be aware of in terms of the action potential characteristic is that the action potential is an all or none response. What do I mean by all or none response? Is that the action potential has a specific amplitude. It doesn't go up. So if I did receive a threshold stimulus or a stimulus stronger than a threshold, I will create an action potential that will propagate along the length of my axon or nerve fiber. What if you've got a much stronger stimulus? You're not going to be increasing the amplitude of the action potential. So the inside will not become 50 millivolts or 80 millivolts. No. What you're going to do is you're going to form more than one action potential to respond to the stimulus. So the action potential is an all or none, whether you have an action potential or not having an action potential, but you do not increase the amplitude of the action potential. How would I be able to respond to a stronger stimulus? So if it's too hot outside, it's, let's say, 80 degrees. Is it different than 110 degrees outside? Yes, definitely. They are different, and it's not the same sensation. So what makes them different? It's the number of action potentials that you're going to be sending from those thermoreceptors in your skin. So your brain is going to be waiting for the signal. So let's say I received 3 action potentials from my thermoreceptors. This means this is the amount of heat I'm exposed to. If my brain is receiving 8 action potentials from the thermoreceptors, this means this is the amount of heat it gets, your body is exposed to. So your brain is going to be receiving those signals and will be determining the intensity of the stimulus according to how many electrical signals have you sent to the brain. All right, it's the same stimulus, it's the same stimulus. So just one stimulus each time, but a stronger stimulus. Like, how would I be able to differentiate the different loudness of a sound? The same exact concept. I have a receptor, and in response to the sound, I would be sending an action potential. If it's louder, I would be sending more action potentials, and my brain will be translating those number of action potentials into a louder sound. So here is the stimulus. This is the stimulus. Here is the electrical signals. So when I increase the intensity of the stimulus, I am exposed to a stronger stimulation. How would I know that I've got a stronger stimulus if I have the same signals that I will be sending? So it's, think of it this way. How your computer functions. It's a code of 0 and zeros and ones. What zero means? Zero means you're not sending a signal. And what is one means? you are sending an electrical signal. So for example, if I wanted to perceive, let's say, a loud sound, the loud sound is going to be creating more signals that will be sent compared to a softer sound. It's still the same type of signals, but the number of signals that I will be sending, the frequency of those signals is going to be lower. All right. And your CPU, your processor, is the one who's going to be decoding the signal on here into this sound, and this signal or this set of signals into a louder sound. Does this make sense? All right, so if I'm using a device, it's emitting sounds, it's emitting colors, it's showing a video, it's showing a presentation, all those signals are the same. It's an electrical signal, 0, 1, 0, 1, 0, 1. And the processor speed is doing what? The processor is decoding those signals according to the code and their frequency, right? Moving on to the refractory period. So again, we started by negative 70 millivolts. I allowed the influx of sodium. It's my depolarization phase. Remember, during depolarization, the sodium channels are open. And I'm allowing sodium inside the cell, influx of sodium. During repolarization, what I will first do, I close sodium channels, preventing any further influx of sodium. During late depolarization, the sodium channels are already closed and no influx of sodium has taken place. In the same time, I've opened the potassium channels and the potassium now is leaking out. This is again during early repolarization. And this is during late repolarization. During hyperpolarization, some of the potassium channels are still open. So I'm still leaking potassium outside of the cell. And my sodium channels are already closed. So nothing have changed from late repolarization. This is the state of my channels on here. So potassium channel here, during depolarization, potassium channels are closed. All this did take place when you received a threshold stimulus. And in response to the threshold stimulus, what happened? You open sodium channels, you completed the depolarization, And when you complete the depolarization, you close sodium channels and open potassium for the repolarization to take place, and then hyperpolarization takes place. What if I've received another stimulus? If I've received another stimulus while I'm still conducting this electrical signal. So let's say I've received another stimulus while I'm still performing my depolarization. Or I've received another stimulus during early repolarization. Or I've received a stimulus during late repolarization or during hyperpolarization. Would I respond to this stimulus? So let's put numbers on here. So this is stimulus number 1 on here, the one that took place during depolarization. Number 2 is taking place during early repolarization. Number 3 is the one that takes place during late repolarization. Number 4 is the one that takes place during hyperpolarization. So which ones do you think you're going to be responding to out of those 4? Would I be responding to any of them first? Would I respond to all of them, any of them? What do you think? Again, my question, I've received the stimulus on here, and while I'm still creating my action potential, I received other stimuli, 1, 2, 3, and 4. Would I respond to any of them? What do you think? Just a guess. Would I respond to any of them? 1, 2, 3, or 4? Yes, I will respond. Which ones do you think you're going to respond to? Again, those are threshold stimuli. All of them are threshold stimuli. 1 and 3. So how do you respond? Can you remind me? How did we respond to a stimulus? What was my response to a stimulus? I opened sodium channels, right? I opened sodium channels. And when I opened the sodium channels, what happened is depolarization in response to opening those sodium channels. So which one now do you think I will be responding to? Would I be responding to stimulus number 1 if it was a threshold stimulus? Yes or no? I'm dealing with this threshold stimulus. Would I respond to stimulus number 1? So again, how do you respond? You open sodium channels. And what actually is the state of my sodium channels at this point? They are already opened. So can I open an opened door? If a door is already opened, Can you open it while it's open? No. So how do you respond again? You open sodium channels. If they are already opened, would you respond to this stimulus, number one? Yes or no? No, because simply, how do you respond? You open the sodium channel. It's already open. So can I open an open door? You can't. So would you respond to stimulus number one? No way. I can't respond to stimulus number one. How about stimulus number two? The one that I've received while I am closing the sodium channels. What do you think? Would you respond to stimulus number two? Again, stimulus number two is taking place during early repolarization while you're closing the door. Can you open and close the same door in the same exact time? What do you think? So here I am closing the door during early repolarization. And in order for me to respond to a stimulus, what I need to do is to open the door. So can I open and close the door in the same exact time? No, you can't. So would you respond to stimulus number two? No, you can't. No, you can't. Because simply, how do you respond again? You open sodium channels. And during early repolarization, what you're doing is closing the sodium channels. So I can't open while I'm closing. How about in number three? Can I respond to stimulus number three? So what is the state of the sodium channels? Sodium channels are closed. They are closed. Can I open a closed door? Can I open a closed door? Yes, it's already closed. I can't open it. Yes, I can respond, but not with a threshold stimulus. Why? Because let's say on here, I need to get 5 sodium in order for me to depolarize, to reach 30 millivolts. So let's remove the sodium on here. You need to get 5 positive in order to become 30 millivolts. So how many sodium channels would you open in this case if you are going to be depolarizing, having 5 positive charges moving inside the cell? You need to open how many? 5 sodium channels. All right, so a threshold stimulus is going to open 5 sodium channels for me to depolarize. What if I'm telling you here, during repolarization, in the same time, I am losing potassium, I am losing positive. So I am losing, let's say, 3 positive out. So if I open the same 5 channels by a threshold stimulus, I allowed 5 in, while I'm losing 3 out, do you think I will be able to depolarize? Yes or no? So again, my question. In order for you to depolarize, you need to get 5 positive ions, let's say, as a number, just for facilitate. the information, right? 5 positive charges. So it allows 5 sodium in for you to depolarize. This is from a resting state. So if I allowed the same 5, but in the same time, I'm losing 3 positive charges out, I'm losing potassium out, would I be able to reach the depolarization? No. Why? Because I don't have 5 effective positive charge gains. Because the net gain is just 2, and 2 positives are not going to be allowing me to reach the depolarized state. It's not enough. So would I respond to a threshold stimulus in this case? What do you think? A threshold stimulus that opens those 5 sodium channels, would I respond to them? No. If you got a threshold stimulus during late repolarization and during hyperpolarization, you're leaking potassium out, so you will not be responding to a threshold stimulus. But can you respond in general? Yes, you can respond, but not to a threshold stimulus. You need something stronger than a threshold stimulus. Something that allows more sodium channels to open. So something that will allow 8 sodium instead of 5 to move in the cell. Now I will get 5 positive charges inside the cell, and at this point I can reach my depolarization. So a threshold stimulus that allows those 5, it's not going to be enough to cause the excitation of the nerve cell during repolarization and hyperpolarization, because you're leaking potassium out. You need something stronger than a threshold stimulus. Yes, I can respond, but to a stimulus stronger than threshold. How come are you able to respond? Because simply the sodium channels are already closed. And if the sodium channels are already closed, I can open them now. But I need to open more of them to substitute the loss of positive charges to the outside of the cell as I'm leaking potassium out. Does this make sense? All right, so what do we call this phase? The phase during which you won't be responding to any stimulus. I won't be responding to any kind of stimulation. Threshold stronger than threshold, any stimulus, I will not be responding to. It's my absolute refractory period. It's absolute. You won't respond to any stimulus during depolarization and early repolarization. How about during late repolarization? Late repolarization and during hyperpolarization, can I respond? Yes, but not to a threshold stimulus. You need a stronger than threshold stimulus. So we call this is a relative refractory period. It's relative. Yes, I will respond, but to a stronger stimulus, not to a threshold stimulus. I won't respond to a threshold stimulus. Again, why? Because I need to substitute the loss of positively charged ions I am losing, leaking potassium out. So again, what is the refractory period? Refractory period is the time when the membranes will not respond to a stimulus for a brief time following a nerve impulse. Absolute refractory period, the membrane cannot be stimulated. Again, you understand now why I can't be stimulated, because simply you respond to the stimulus by opening sodium channels. And during depolarization, your sodium channels are already opened. And during early repolarization, you actually you're closing the sodium channels. So you will not be stimulated. And relative refractory period is the time when the membrane can be stimulated, but by a higher intensity stimulus, stronger than threshold. Why higher intensity stimulus? Because I need more sodium channels to open to substitute the loss of potassium during late repolarization and during hyperpolarization. So we're looking here, this is my absolute refractory period, takes place during depolarization and early repolarization compared to during late repolarization and hyperpolarization. I have a relative refractory period. So what's going to be determining the electrical conduction velocity? How fast is the electrical signal going to be conducted through the nerve fiber? First factor is going to be the diameter of the nerve fiber. The larger the diameter, the faster the conduction will be taking place. Another very important factor that affects the velocity of the electrical conduction is the state of the myelination of the nerve fiber. Myelinated nerve fibers gonna have a faster electrical conduction speed. So how this works? So again, what is the electrical conduction? You are performing depolarization, repolarization, depolarization, repolarization, depolarization, repolarization, depolarization. And this electrical signal keeps propagating along the length of my nerve fiber until it reaches its effector organ or conducted to another nerve cell. This is what happens in an unmyelinated nerve fiber. If we're looking on here to the myelinated nerve fibers, remember they are covered with myelin sheath formed by the Schwann cells in the peripheral nervous system. And remember, those membranes are going to be insulating the electrical conduction. So the signals going to be formed only in the nodes of Ranvier. Depolarization, repolarization is the opening and closure of those channels takes place only at the nodes of Ranvier. So as if the electrical signal is jumping from a node to the next. So which one is faster, do you think? If you are forming an electrical signal at every single point of the nerve fiber, like this, or if you're forming the electrical signal only at the nodes of Ranvier, which one was faster? Unmyelinated or myelinated? Which one is faster? Follow here the pointer. This is the conduction of the signal on here in an unmyelinated nerve fiber. And now the conduction in myelinated nerve fiber. Which one was faster? So again, this is unmyelinated. Check the time. Start your timer now. Or myelinated. Which one was faster? Myelinated. You're skipping parts of the axon. You don't have to open and close the channels in. So this saves you a lot of time. You are jumping from one node to the next node. So it's going to be much faster electrical conduction. So again, which one going to have a faster electrical conduction? Myelinated or unmyelinated axons? My myelinated axons are the ones that will have a much faster speed of electrical conduction. All right, we can have a break for 5. We come back, discuss the synapse between the neurons. Any questions before we leave for a break? Any questions?